Assalamu alaikum. In this video we will talk about the possible drug targets for the treatment of novel coronavirus disease. For the development of any drug, it is necessary to identify some drug targets. Normally, during signaling process, the angiotensin 1 produced from angiotensinogen by action of renin follows two pathways. Angiotensin 1 binds with ACE2 receptor and produce proteins involved in tissue protection or protect lung tissue from injury. While angiotensin 1 also interacts with ACE receptor and results in tissue injury in case of any viral infection to kill infected cells. When SARS coronavirus 2 enters in the body, the spike proteins of SARS coronavirus 2 binds with binding site of receptor ACE2 and cellular proteases prime these spike proteins, and the virus enters in host cell after these interactions. After entry into the host cell, the virus starts viral multiplication. The ACE2 and the proteases have cellular potential for infection. Due to their involvement in infection, these two components can be used as potential drug targets for the development of drug to stop infection. So, spike protein based vaccines can be developed. These vaccines may produce antibodies that can interact with spike proteins and stop infection. The priming activity of cellular proteases can be inhibited by developing some cellular proteases inhibitors. These cellular protease inhibitors can prohibit the interaction between ACE2 receptor and the spike proteins by stopping the priming of spike proteins. The ACE2 receptors can be blocked by developing ACE2 antibodies. That antibodies may block interaction of virus with ACE2 receptors. While another method can be used to develop some therapy developing soluble ACE2. These soluble ACE2 may competitively bind with spike proteins and reduce interaction of these viruses with ACE2 receptor, and it may regulate tissue protection phenomena to protect lung from injury in case of infection.